Once the plan is set, each system will undergo dedicated verification plan. Solution, such as upstream verification solutions or downstream verification solutions. Each verification plan will be clearly defined in detail. Which devices in these verification solutions need to be verified? These devices are what kind of pollutants they contain, and what they dirty holding. How long is the duration, and then what cleaning methods are used to carry it out? Finally, what sampling methods do we use to confirm that it meets the requirements? What are the acceptance criteria? And all these questions will be in your protocol. It is clearly defined inside. After the protocol is approved, the next step is to execute it on site. Protocol. During execution, there will be some dirty holding. Clean the CIP, then take samples after cleaning, and after the test report is out, CIP. Then confirm whether it is qualified. Of course, this also involves clean holding. After cleaning, there's also a study on clean holding. Clean holding study. So this is when it comes to the entire cleaning validation. It might involve some dirty holding, clean holding, and also includes some aspects of the sampling method itself. Clean holding. Certain. To form a clean verification report. Then, after completing all the cleaning systems, there is a summary report. This is basically the entire cleaning verification process. Now, let's talk more specifically about the main types of pollutants, including our detection methods and methods for detecting these pollutants. First, some are mainly organic solvents, or organic substances. Organic matter, which includes solutions of certain proteins. This includes some media, as well as upstream and downstream or downstream commission-based products media. And some media that might not have run out. Media. These things, in our large molecular terms. We use the TUC method to detect these organic residues. TUC. Why use TUC's method instead of protein-specific methods? TUC? Because during the CIP process, we generally use strong acids or strong acid CIP. Alkalis, and also include alkaline solutions like CIP 100 for cleaning. CIP 100. Proteins are very prone to degradation or breakage in this alkaline solution, and strong acids and bases cracked into fragments. So if you measure some of the original protein concentration or content, it no longer makes sense, because it has already broken into some fragments. So for large molecules, this is different from small molecules. Small molecules can be measured for some purposes, such as HPLC to measure the content of specific compounds, HPLC. But for large molecules, the content of TUC is basically all tested, TUC. Using TUC methods to detect proteins or residues in all organic matter, TUC. Stay. Then, for inorganic substances, such as some inorganic salts. It also includes sodium hydroxide, mainly used to measure the TUC. Conductivity testing methods, TUC. Measurement. There are also some things, like our cleaners. It's CIP 100, which is a mixture. CIP 100. It may also include organic melting machines and some inorganic salts. Therefore, it is necessary to measure conductivity and TUC. TUC. Besides this, some other tests are needed, such as testing for microorganisms and endotoxins. We just talked about some testing, so now let's talk about our sampling methods. Sampling methods in cleaning verification mainly include three types. The first is rinse, which mainly serves as rinse water. Rinse. Some samples of rinse water can be quite representative and can moisten most of the equipment. Some surfaces, which can be exposed to a large number of surfaces. Rush forward, sampling is more representative. The main thing here is that the rinse water mainly tests some of its conductivity, biburden, and detoxin TUC. Besides this, there's also visual inspection. This is also very critical. Before sampling, or before swapping, swap. It will first check if the equipment is clean. Confirm this visual, including looking at some of its positions. Visual. How do you look? Right. What is the acceptance standard? Visual inspection is also required. Visual inspection. Besides these, there are some swaps, which are some poor samples. Swap. Poor test sampling is actually mainly focused on some worst cases. Worst case. Some points. So, which are some of the points in the poor test sampling compared to the worst case? Worst case? Here are some of the main options for worst case. Worst case. Principle. First, for example, the hardest places to clean, and also the areas where products tend to accumulate most easily. And some products that dry out easily or are easily absorbed. There are also changes in pressure, which gradually decrease from high to small. Or suddenly the place that goes from small to large to close. These are places where products tend to accumulate. For areas where these products tend to accumulate, we need to conduct some poor sampling tests. Go prove that the things you accumulated there can also be cleaned clean. So, which devices need to be cleaned and verified? Roughly, we can divide it into two parts. First, this device is reusable. Second, the equipment comes into direct contact with the product. For disposable products, we do not make disposable products. For example, discarding items like disposable. There is no need to perform cleaning verification because it will not be used for the next product. There are also some who don't directly interact with the products. There's no need for cleaning verification either. I think this is easier to understand. Next, we gave some examples. Mainly, regarding the biological data of macromolecules, there are certain devices that perform cleaning verification. For example, upstream, mainly DF skid, harvest tank, transfer PIP, ING, and some centrifuge, centrifuge. Mainly some scattered machines. These are some of the main upstream equipment. For downstream users, most of the equipment is VPE. Arc top process, DF. VF, UFDF, these devices. VF, UFDF. There are also some stainless steel cans and some stainless steel holding. Tank, transfer piping. There are also some media systems and buffer systems, media buffer. Preparation tank holding. Tank, and some corresponding papping methods. Tank, papping. There are also some cleaning machines and small parts cleaning machines. There are also small parts cleaning machines available upstream and downstream. These are mainly devices used in cleaning verification. Now, after discussing the second stage, the verification of the cleaning process. Entering the third stage is the confirmation of ongoing cleaning processes. After completing the PPQ stage, we enter the third stage. PPQ. The goal of the third phase is to continuously monitor our cleaning process while it remains under control. In the state of verification. So what is the main approach? First, we will have a plan to confirm ongoing cleaning. Plan. In this case, we call it CCVP. CCVP. In this CCVP, we define some sampling frequencies, CCVP, including the sampling sites. Generally, CCVP is held every year. CCVP. And if it involves some special sampling, this will also be clarified in the CCVP. CCVP. Second, depending on the specific batch or plan of the CCVP plan. Batch sampling, then on-site cleaning and confirmation. Then collect some cleaning confirmation data in this area. Detected data. Based on this data, we conduct annual analyses of these cleanliness confirmation figures. This also includes whether there are any deviations or changes. Then the data is analyzed to see if the cleaning process is still under control. Are there any signs of deterioration? Some of the main purposes or primary tasks of this third stage are to look at our cleaners. Is the art still under control? In the second part, I want to talk about the specific process during the product transition. How should we control products and control some of the public equipment? Prevent some crossovers. First, here we also share something called decision tree. To put it simply, we use product evaluation to confirm exactly what we should do. Once this production line or these devices complete the auditory verification of some commercial products. Later, during the process of introducing any new product, we all need to conduct some NPI, which is a risk assessment for introducing new products. NPI. In this assessment, we identify which devices need to be shared. Then, based on these shared devices, check the cleaning capabilities of these products in these shared devices. How is it? This may involve products, and some of these devices have different materials. If this product is made of different materials, its cleaning power may vary. Without data on cleaning capability in this area, some tests are needed, such as doing coupons in the lab, coupon study to verify that on different materials, study. How is its cleaning ability? Based on the results of these coupon studies, coupon study. Compare it with the results of the worst case we've already verified. Worst case. If its cleaning ability is no worse than the worst case we've already completed, worst case. So we just do the usual cleaning checks and spray some water. That's enough. If it's worse than the worst cases we've already verified, worst case. For clinical non-PPQ projects, PPQ. Enhanced clarification. This means intensive cleaning confirmation, including spraying some water and some sampling. If it's the 3-leaf or PPQ stage, we will directly perform cleaning verification. PPQ. Why is a cleanliness confirmation done clinically? How about strengthening cleaning and confirmation? This is because in clinical sample growth, there may not necessarily be 3 consecutive batches or so. Such a process may only be done 1 batch or 2 batches. This way, it cannot complete a clean verification. So we use some strategies for enhanced cleaning confirmation to do this. Although this isn't called clean verification, some of its sampling sites. Some assessment methods are basically consistent with cleaning verification. Next, let's talk about how to control the product transition process. This includes conducting some risk assessments. At the NPI stage, some products like this are already excluded. NPI. Additionally, among some shared equipment, we need to distinguish which ones are in direct contact with the product. Some shared equipment should be clearly listed. Then we conducted some coupon studies, coupon study. Check the coupon results to confirm whether it's for cleaning verification, coupon, enhanced cleaning verification, or clean verification. Of course, during product switchovers, we also need to recalculate some of its cleaning limits to guide it. Product switching. This cleaning limit mainly refers to the TOC limit. TOC. We mainly rely on 3 methods. One is the PDE method, PDE. One method is the minimum liquid accumulation volume at 1/0000 and 10 ppm. 10 ppm. Using these 3 methods to calculate 1 centimeter each. Then take the smallest centimeter as the centimeter for cleaning. Execute during product switching. This ensures that during product switching, the previous batch won't be switched to the next. This leads to cross-contamination, including conductivity, by burden. Endotoxin is basically required to be implemented according to WFI standards. Endotoxin, WFI. Then, during the cleaning evaluation during product switchover, we also output some information. After this assessment, we will provide some information on whether to confirm cleanliness or perform cleanliness inspection. Certificate. At the same time, you should also know whether some of the elastomers need to be replaced. There are still areas where removal, or some elastomers that can be reduced. These are all based on some validations we have already completed. Its elastomer then outputs the specified output based on the library's results. Confirm whether we need to perform this action. This helps maintain our overall production rhythm, including some product switching in our production. The efficiency of some actions needed to be performed at that time. Including the improvement, you can save a lot of money. Next, let's talk about regulations regarding cleanliness verification and some requirements in this regard. Why do cleaning verification be necessary? I think there are probably two reasons for this. First, prevent cross-contamination to ensure safety during use. Additionally, we hope that many producers. They also hope to share and contribute to production, improving equipment utilization. I think most pharmaceutical companies or manufacturers nowadays don't have a single production line, or a single product, or even just one product. They hope that as long as they don't violate certain rules, such as the non-compliant contributions mentioned by the National Medical Products Administration. For some products, we hope to maximize the possible contribution of the product. This improves equipment utilization. This is where some issues are mentioned, such as the GMP 2010 section of the Drug Administration GMP 2010. These products need to be separated, requiring dedicated production lines, including some guidelines from the EU's GMP. It also mentions that when designing some maternity room facilities, it is necessary to consider the product to prevent some cross-contamination. From a risk perspective, efforts should be made to minimize the risk of cross-contamination of products during production. Then there are some regulations and guidelines for some cleaning validation guidelines. Some demands. First, some GMPs in various countries specify how to verify cleanliness, GMP. Why do we do it? It sets very clear requirements. For example, the EU GMP Appendix, Confirmation and Verification, GMP. Both the NMPA and GMP 2010 editions mention certain requirements for cleanliness verification. NMPA GMP 2010. FDA certifications include cleanliness validation, such as some inspections FDA inspection. Requirements. Also, the EU has mentioned how to establish and set it up and some people-based ways to contribute to production. Some limits and settings for some people in the health space. Then, the TR29 PDA was cleaned and verified. DATR 29. Then, including some TR49 from biologics, TR49. Some points to consider for biopharmaceutical cleaning validation. Also, the ISP's cleanliness verifies the life cycle. ISP life cycle. These are some of the current regulatory requirements for cleaning verification. I won't list or elaborate on them one by one here. Next, I'll share a case study of how we conduct cleaning validation. Or in some of the case studies we have done. We cited a clean verification that makes good use of process equipment. First, in making good use of process equipment, we will make good use of process equipment. List all the devices that will be shared. For example, one is a centrifuge. The centrifuge used, including some pipelines of the centrifuge, including some pumps, including some hobstacks and some transport platforms hobstack, transport. Some piping, listing all the devices that will be used. Piping. At the same time, these devices are grouped based on certain standards, divided into some equivalents or similarities, focusing on equivalence concepts. Within these groups, they can run each of these 3 devices once, group. Or one device can do it 3 times. These are some equivalent devices. If it's for similar devices, will be the biggest or worst case. Do it 3 times, then just do the middle one once. So, based on these assessments, we decide which things need to be done in 3 runs, which need to be done to reduce some of the cleaning verification workload. Run. The verification method was just mentioned. First, after cleaning is complete, there is a dirty holding time. Dirty holding. The dirty holding time is mainly brought up from the production side. Dirty holding. Production is based on some of its bowel movement needs, including its resources. Give a list of how long they might hold from the end of production to cleaning, holding. Only then can you arrange the time. So, this will produce the biggest dirty holding time. For example, here, after the centrifuge is produced, the largest holding time is about 8 hours. Holding 8. There are also some bumps in tanks, bump tank. The maximum time may be 72 hours. 72. Based on these, we will challenge them as some corresponding ones. After cleaning here, some rinse samples are taken, rinse. Visual inspection. Then swap the scene. Swap. Because a swap was done, the device will be recleated swap CLE. And let it return to a clean state. Then accept the standard. Some standards for visual inspection are visual inspection. We needed to look at the surface to see that there are no residues of any products. There are no superficial issues. As for some TLC limit calculations, which I just mentioned, TLC. You need a 1/000 th of a diary material and 4 ppm, including PDE data. PDE. This calculation uses 3 methods, taking the lowest value. Then, conductivity, by burden. Antitoxins, which can be customized according to process requirements antitoxin. However, many times it is only WFI standards that are selected. WFI. However, this can also be tailored according to process requirements. Then, after the final product verification, there will be a result. There's an output item listed here which shows how much we've challenged. For example, the most low means you have to challenge dirty holding for 8 hours, dirty holding. Our challenge duration must be longer than 8 hours. Here, let's list it out. For example, the shortest time among 3 runs is 10 hours 0 run. 9 minutes. But when outputting the SOP, we need to set it not greater than SOP. Less than or equal to 8 hours. The following 72 hours are the same. During the challenge, cleaning verification must be longer than 72 hours. However, the final SOP output is less than or equal to 72 hours. SOP. Completing cleaning verification requires 3 consecutive successful batches. Verification complete. At the same time, each of these tests must pass its visual inspection to qualify. The entire CIP program is fully capable of running. CIP. Including the overall test results, TOC, conductivity, TOC. Both biburton and antitoxin meet the established standards. Biburton antitoxin. It is based on these factors that we can declare that this cleaning verification is qualified. Alright, that's all I have to say today. Let's see how the Q&A is now. Q&A.